Let's continue our conversation of DNA by talking about how chromosomes are structured and arranged within the nucleus or the nucleoid. So we're going to start with some keywords and then a brief overview of the chapter. Just a couple choice keywords to help you understand what's going on. So first one is this protein encoding gene or structural genes. So we have a DNA, we have a segment of DNA, which is our gene. It can code for a protein like we're used to, so DNA, mRNA, protein, or it can code for a non-coding RNA. Remember, it exists a long non-coding RNA, so it's not a protein encoding gene, but an RNA encoding gene. There's also those for tRNA, rRNA, and a host of other things we'll talk about. The next word is this intergenic region. So it is as it sounds. So you have one gene, two genes, and it's the area in between those genes. We have the origin of replication. Now this is where replication will start in bacteria and eukaryotes. You'll notice that bacteria tend to have one. So you have an origin of replication. This is where we're going to open up our DNA, and replication is going to go bidirectional. Bacteria tend to have only one. They only have one chromosome, so it doesn't take as long to replicate. Whereas in eukaryotes, they have many and they're linear, so they can have multiple origins of replication. So at each one of these origins, you can open up the DNA and start to replicate it. We have repetitive sequences. So we see GGAAT, 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 so on and so forth. Those are repetitive sequences. Now, we can classify these as highly repetitive and moderately repetitive. So if they have few hundred to several thousand, they are moderately repetitive. I'm sorry, there's a typo here. For highly repetitive, they have to be tens of thousands to even millions, okay? Then there's also unique sequences or non-repetitive sequences that you see once or relatively a few times, like maybe 1 to 5 times or so. We have this thing called microdomains. So when we're looking at the prokaryotic DNA molecule here, each one of these lines is a strand of DNA, a part of the chromosome. Those are considered the microdomains. Now this shape, what we're visualizing here, is possible by these NAPs or these nucleoid associated proteins that allow them to loop and be held into place. Next, we have this idea of supercoiling. So supercoiling is adding these twists and turns into our DNA molecule. Sometimes we have enzymes, so like DNA gyrase and topoisomerase, whose job it is to either introduce supercoiling or relax supercoiling. And it's specific to if it's positive or negative. And even when we are trying to unwind the DNA, as when we're doing replication, we can introduce supercoiling ahead of our replication fork. And now we need a topoisomerase to come in and relax that supercoiling. And this occurs in both bacteria and eukaryotes. Now, if we look at our chromosome, okay, we have introns and exons. So introns tend to be those non-coding regions, almost like the intergenic region, although they can occur within a gene. So these are non-coding regions within a gene that gets spliced out. And then you have exons, which are the coding regions or the express regions. We have a centromere. This is a DNA molecule found usually around the center of a chromosome, depending on the type of chromosome. We have our telomeres that's found at the end. Those are made of repetitive sequences. Now, our kinetochord is a group of proteins that attach to the centromere, okay? So the centromere is DNA, protein binding DNA, essentially. The kinetochord is a group of proteins that attach to that centromere, and then our microtubules emanate out of it during cell division. Now we have this idea of transposable elements or transposons. These are the so-called jumping genes. What that means is they can insert and excise themselves out of the chromosome. So there are certain places that they can integrate, and then when they want, they can excise out and go integrate somewhere else. So they can move around our genome, and we'll talk about this more in detail in an upcoming video. Okay, so those are just a couple keywords to introduce you to some things that might not be a review to you. So these are some things that you might be hearing for the first time. And I didn't go through all the words, so some of them will be explained in more detail in the text. So now, in general, what is the primary purpose of our genetic material? I think everybody should be screaming out an answer now, right? So you should be telling me that the purpose of our genome is to what? We need to store the information. It needs to serve as a blueprint, right? We remember that. And we need to be able to pass it on. So it codes for our proteins. That's the storing information. It gives us the blueprint or dictates what we look like, and we pass it on to our offspring, or it can be inherited. So these chromosomal regions have to have certain processes in place. One is synthesize. So we have to be able to take these chromosomes and synthesize RNA proteins from them. We need to be able to replicate our chromosomes. They need to be able to segregate during cell division, and we need to be able to compact them inside of our nucleus. So like you'll see in subsequent videos, there is over 6 feet of DNA that has to fit into a microscopic cell. How is that possible? We can't just be pushing and jumbling it all in. We have to neatly be able to arrange this. So there are several steps we'll take to compact it to make sure that it is safe. Okay? And now you'll see that you have a chart included in your notes that you should be able to fill out. Okay? So make sure that you take a look at this, and after reviewing videos and reading the chapters, put everything aside and see if you can fill this in. See if you got anything from what you were reading and learning. Okay? All right, so that's it for this video. Tune in to the next one. We'll talk about bacteria. We'll talk about eukaryotes, and there's one on transposons. See you soon.